Hello there, and welcome to Teach Me in 10, where we break down complex science topics in under 10 minutes. Today, we're diving into the fascinating world of DNA methylation analysis with dPCR, guided by Zahid Ahmed, a senior support scientist at Roche Sequencing and Life Science. Zahid will be simplifying this cutting edge technology and explaining how it's revolutionizing our understanding of genetics. So, without further ado, let's jump straight on into it.
Thank you for the introduction. The topic I'll present is quantification of DNA methylation using methylation-sensitive restriction enzymes and digital PCR. First, the background of this study will be presented. DNA methylation is an important epigenetic mechanism used by cells to control gene expression and tissue differentiation. In addition, it plays a critical role in onset and progression of many diseases, such as in cancer and aging. DNA methylation often occurs as cytosine molecules within the CpG, cytosine phosphate guanine, dinucleotide sequence, and is characterized by the presence of the signature 5-methylcytosine without altering the sequence of DNA. Currently, sodium bisulfite treatment prior to PCR is a common method to determine the methylation status of genomic sequences. Bisulfite conversion approaches are not compatible with the uracil DNA glycosylase UNG PCR system, such as the Roche digital life cycler. UNG is often utilized to eliminate carryover contamination. When using bisulfite conversion, it may degrade DNA samples, leading to poor conversion efficiency and conversion specificity. During sodium bisulfite treatment, unmethylated cytosine residues indicated as UM in gray are converted to uracil, whereas 5-methylcytosine indicated as M in green remain unaffected. During PCR amplification, uracil residues are converted to thymine. A UNG system digests uracil containing DNA fragments, thus no PCR product is detected. An alternative efficient approach is to use methylation-sensitive restriction enzymes, MSREs, to analyze the methylation status of cytosine residues in CpG sequences. These restriction enzymes are not able to cleave methylated cytosine residues, leaving methylated DNA intact. There are different MSREs, such as BSTU1 and HHA1, that can specifically cleave unmethylated CpG dinucleotides, resulting in the preservation of methylated DNA fragments. Digital PCR is an innovative method for performing absolute quantification. It does not require real-time detection, relying instead on a straightforward endpoint analysis. Initially, a specific DNA master mix must be prepared, which includes the necessary primers and probes. Then the sample is divided into thousands of partitions. After the reaction is finished, the partitions are counted. Positive partitions show high level of fluorescence, whereas negative partitions do not. In the end, Poisson distribution predicts the proportion of partitions containing a given number of target sequences. A higher precision, sensitivity, and accuracy can be achieved with digital PCR, since interference of PCR inhibitors and competing background DNA is reduced by partitioning of the reaction mixtures. When using digital PCR technology for DNA methylation, there are several benefits, such as no need for standard curves. dPCR quantification is more accurate since it relies on clustering partitions into negative and positive groups, and then calculation of the target concentration. In addition, there is higher precision. The more partitions the system can handle, the greater the precision. Also, there is increased signal-to-noise ratio. Partitioning effectively enriches target concentrations in target-positive partitions, relative to those of potential PCR inhibitors and interfering background sequences. Next, DNA methylation analysis will be mentioned. The target of interest in this study is Ras family 1 isoform A, Ras F1A, which is a well-known tumor suppressor gene that is apparently methylated in many human cancers. Hypermethylation of the Ras F1A promoter CpG island silences expression of the gene in many cancers, such as lung, breast, and neuroblastoma. The CpG regions in Ras F1A extend from the promoter to exon regions. Primers and probes were designed to specifically target the promoter region. During initial experiments, only 30% of quantification was detected in CP genome healthy human methylated DNA samples, and 15% in methylated DNA positive control. After investigation, it was found that CpG regions contain a high GC content, about 72.4% in this study, which led to lower quantification. To mitigate the low quantification issue, high GC enhancers such as DMSO, glycerol were tested using different concentrations shown in this plot to the left. The x-axis shows a healthy gDNA and methylated DNA along with different concentrations of enhancers. Enhancers such as 5% and 7.5% DMSO significantly improve accuracy or quantification, with expected quantifications falling under 95% confidence intervals. To the right, the 1D clustering shows positive clusters in red and negative clusters in blue. The suitable concentration of DMSO is within specific range. Using 10% DMSO can start to interfere with performance. A target-specific TaqMan multiplex dPCR assay was developed for detection of Ras F1A promoter methylation. The HHA1 restriction enzyme was used in this study. The target gene Ras F1A was used along with reference 1. Actin gene was used for the purpose of monitoring digestive completion. And reference 2 EIF5 gene was used for input correction DNA. The dashed line shows the location of HHA1 cleavage sites on the PCR amplicon region of the Ras F1A promoter, and the solid lines indicate the location of cleavage sites on reference 1. On the right, the 1D clustering shows healthy genomic DNA and methylated DNA before and after restriction enzyme digestion. HHA1 digests unmethylated cytosines on reference 1 actin and Ras F1A by showing no formation or very few positive clusters in red. Reference 2 EIF5 shows positive clusters in each well of genomic DNA and methylated DNA, since it has no cutting sites for restriction enzyme. Thus, methylated cytosines on the target gene Ras F1A resist enzyme digestion and show positive clusters or PCR amplification. The methylated fraction data analysis helps determine the percent of methylation. This shows 2D clustering of target and input correction control as an example. The ratio of the target gene Ras F1A and reference gene EIF5 is shown, with the HHA1 digestion ratio of 0.975 and without digestion ratio of 1.004. The second step is to find the percent fraction of methylation. For this example, 97% of the sample is methylated. With the Roche digital light cycler, 3D clustering, which is important for data visualization, can be obtained from 3 of the fluorescent channels used in the study. Each cluster is color-coded to better distinguish the clusters. On the left, the methylated DNA positive control after enzyme digestion is shown. To the right, healthy human genomic DNA is shown, which shows less clusters in the FAM and HEX channel due to the enzyme digestion. Lastly, the methylation status was measured in various lung cancer and breast cancer cell lines and in healthy human genomic DNA. On the left, methylation status of the Ras F1A promoter region is shown. In the healthy genomic DNA sample, the methylation status was less than 0.7%. Different amounts of methylation levels were detected for cancer cell lines depicted. In the exon 1 region to the right, different levels of methylation could be detected using the same cancer cell lines. Other studies, including this one, observed different methylation status of RAS F1A between the promoter and exon 1 region. Thank you for watching.
Thanks so much for tuning in to this Teach Me in 10 episode, and I'll see you again very soon.